Welcome to this explainer. Okay, so have you ever stopped to wonder why you don't just, spontaneously combust, or why digesting your lunch doesn't take a million years? Today, we're popping the hood on human biochemistry to demystify these microscopic, highly engineered little machines that keep our bodies running. If you've ever wondered how biological life is literally even possible at a molecular level, well, you're absolutely in the right place.
So, let's start with a really fundamental question. The ultimate problem of life, really. How does a single cell perform millions, literally millions, of reactions in just a fraction of a second? The reality is, chemical reactions on their own, they are agonizingly slow. Without a specialized biological accelerator to sort of bypass the waiting game, life as we know it would just grind to a halt.
All right, let's dive right in. The biological accelerators we're talking about here are called enzymes. These are highly specific globular proteins that dramatically speed up biochemical reactions without actually getting consumed in the process. Think of them as the ultimate reusable spark plugs. Most of these engines are just floating freely in the cell's cytoplasm, though it's worth noting some of the prokaryotic respiratory ones act like they're bolted down, firmly anchored to the plasma membrane.
Here's our blueprint for today's teardown. First, we'll look at the architecture of an enzyme. Second, the catalytic cycle. Third, environmental speed limits. Fourth, system overrides. And finally, the master regulators. Let's get to it.
Section 1. Architecture of an enzyme. Inspecting the parts of the machine.
Now, while some simpler enzymes are made purely of protein, many of the really crucial ones are what we call hollow enzymes. You can kind of think of a hollow enzyme like a completely functional engine unit. It needs a protein base, that's the apoenzyme, or basically your engine block, plus a non-protein plug-in, which we call a cofactor, to actually turn the ignition.
Looking at the anatomy of this engine, there are a few different types of these plugins needed to get that apoenzyme running. Sometimes they're just loosely bound inorganic activators like magnesium ions. Other times they're organic coenzymes, which, fun fact, are derived from the vitamins in your diet. And sometimes they're tightly bound prosthetic groups like heme, physically locked right into the structure to make the whole machine work.
To understand how this biological engine actually docks with its fuel, we have to look at the active site. This is a 3D charge-bearing cavity. Historically, we've compared Fisher's lock and key model, which says the enzyme is perfectly, inflexibly shaped for its substrate right from the start, against Kochlin's flexible induced fit model.
Now, modern biochemistry overwhelmingly favors the induced fit model. The binding literally induces a physical, conformational change. Basically, the enzyme molds itself right around the substrate to align the catalytic groups perfectly. So it's less like a rigid metal keyhole and much more like a precision baseball glove wrapping tightly around a ball.
Moving on to Section 2, the catalytic cycle, the engine in motion.
We can actually break this engine's stroke cycle down into 4 precise mechanical phases. Step 1, locking the substrate fuel in. Step 2, shifting its shape in that induced fit we just talked about. Step 3, forcing the actual product formation at the active site. And Step 4, releasing that product, so the unmodified enzyme can hit reset and fire all over again. Here, the enzyme acts as pure orienting architecture. By grabbing the substrate and holding it in the exact, perfect position, it drastically lowers the required activation energy. It's essentially carving a high-speed tunnel straight through a chemical energy mountain that the reaction would normally have to climb completely over. But here's the crucial point. This engine definitely has a top speed. We map this out using Michaelis-Menten kinetics, and we call this absolute top speed, Vmax. Once all the active sites are totally saturated with substrate, the rate just plateaus. Adding more fuel at that point produces absolutely zero increase in the reaction rate, simply because every single machine on the factory floor is already running at max capacity.
Which brings us to Section 3, environmental speed limits, operating thresholds, and red lines.
You see, each of these engines is exquisitely tuned for its specific local environment, and that's because they rely on the ideal ionization of their amino acid side chains. Just look at this variability. Pepsin, for example, thrives in the highly acidic, burning environment of your stomach at a pH of around 2. Meanwhile, pancreatic lipase, it requires a much gentler, alkaline pH of 9 to do its job in your intestines. Pushing an enzyme outside of its optimum peak and into one of these denaturation hazard zones physically disrupts its ionic and hydrogen bonds. And to be clear, it doesn't just slow the machine down, it completely destroys its three-dimensional shape, rendering it completely, totally useless.
We see a very similar but honestly even more dramatic effect with temperature. Thermally, the reaction rate roughly doubles for every 10-degree rise, climbing right up to the human optimum of 37 degrees Celsius. But pushing it further, it doesn't give you a gentle downward curve. No, it drops you off a literal hot zone cliff. Excess heat irreversibly melts the enzyme's molecular bonds, permanently destroying the machine.
Next up is Section 4, System Overrides, putting the brakes on metabolism. So how does the body actually pump the brakes? The primary way it exerts control is through inhibition. This is a regulatory process where specific molecules bind to the enzyme to decrease its reaction rate. It is essentially throwing a physical wrench straight into the turning gears of our molecular engine to force it to power down.
And the way this block happens is absolutely fascinating. Reversible inhibitors form weak, temporary bonds. They pause the reaction and then let go. But irreversible inhibitors? They permanently bind and physically shatter the enzyme's architecture. This destructive takeover isn't just a fun theory either. It's the exact clinical mechanism of how the drug penicillin kills bacteria.
When the body does this naturally, it uses two main tactics. First is competitive inhibition, where a structural analog literally crams itself right into the active site, blocking the real fuel. Think of malonic acid inhibiting succinate dehydrogenase. The second tactic is non-competitive inhibition. Here, a molecule binds to a secondary allosteric site, which sends a shockwave through the protein that distorts the main active site's shape. This effectively turns the machine off without ever even touching the primary docking port. Finally, Section 5, Master Regulators, the interconnected factory floor.
Across this massive cellular factory, we find incredibly specialized classes of these machines. We have zymogens, like pepsinogen, which are manufactured and secreted in a locked, inactive state, so your stomach doesn't, accidentally digest itself. We have oxidoreductases, which specialize in hot potatoing electrons down the respiratory chain, and we even have ribozymes. These are rare RNA molecules that break the whole only proteins can be enzymes rule to perform catalysis themselves.
Together, these individual molecular machines network to provide really precise, dynamic control over complex metabolic pathways. Through intricate switches, strict environmental bounds, and rigid structural specificity, they make absolutely sure the chemical traffic flows perfectly across the entire cell map. So as we wrap up this explainer, I want to leave you with this thought to take away. Enzymes are not merely passive chemical accelerators. No, they are the ultimate control centers of life itself. Every single second you are alive is mediated by these precise structural shifts, which leaves us to wonder, with billions of these reactions happening in perfect sequence right now, what happens to the entire interconnected factory when just one of these tiny microscopic machines breaks down? Thanks for joining me on this teardown, and keep questioning the incredible mechanics of life.